Hi, everyone. We're going to go ahead and get started now. I want to welcome everyone to our webinar on comprehensive variant detection in pediatric leukemia research. And we're excited that you've decided to join us today. I'd like to give a brief introduction of our speakers. I'm Alex Sackel. I'm the segment manager for cancer genomics here at PacBio, and I will be your host today. I'm honored to be joined by two of our collaborators who are really pioneering the use of accurate long-read whole genome sequencing to uncover driver mutations in pediatric leukemia samples, particularly those that were missed by other technologies. So I'm going to first give a quick overview of the cancer genomics applications that we're enabling at PacBio, and then we'll play a pre-recorded talk from our speakers. But I will tell you they are both on the line with us and will be answering questions live during the Q&A. Just a couple of housekeeping items. First, due to the number of people calling in, we've muted all the attendees. We are recording this webinar and we'll make it available in the next week or so please be on the lookout for a follow-up email. The presentations we have will be followed by a question and answer session. You're welcome to submit your questions during the talks by typing them into the section of the control panel over here. It's the box with a question mark, and we will address them during the Q&A. And we've also, on the tab next to that, uploaded a number of complementary pieces of literature. So please download those and take them with you. And then finally, there'll be a survey at the end, and we ask that you please take the time to fill it out. Okay, that's all the housekeeping, so I'll jump right in. I'm excited to tell you today about how accurate long-read sequencing with PacBio HiFi is already enabling the next generation of cancer genomics by providing comprehensive and multi-omic characterization of the cancer genome, epigenome, and transcriptome. So first, cancer is a disease of the genome, but much of the effects of these mutations are expressed at the level of the transcriptome and epigenome. This means that to really understand cancer, we need to be able to perform comprehensive and multi-omic profiling of these somatic changes. With accurate long reads from PacBio HiFi, you're able to see the complete cancer genome, so that's what we're showing on the left here in blue, which includes not only small variants like SNVs and indels, but also structural variants, phasing, and direct detection of methylation. And we also enable the researchers to look at the complete cancer transcriptome in magenta, which goes beyond simple gene expression to include detection of isoforms, fusions, and expressed mutations, which is directly tied to things like neoantigen discovery. So we're going to start with the transcriptome here, since most of the rest of the talk will be focused on the genome. I'm going to walk through two quick examples where HiFi enabled detection of novel isoforms and fusions in both bulk and single-cell RNA sequencing. So first, why are long reads so much better at reconstructing the transcriptome? On the left in the colored diagram, you can see the exons of the gene shown in pink, and below it are the multiple mRNA isoforms that are being produced. The challenge with short reads is that you only identify the specific isoforms if you happen to have a read that spans a splice junction. So that's what we're showing here with the dashed lines. And it's estimated that short reads are only able to reconstruct about 20 to 40% of the transcriptome. In long read sequencing, on the other hand, shown here on the right, you just read the entire transcript in one go, so you know which isoforms each read came from without having to do any assembly. And because of this, we actually think that the role of differential isoform usage in cancer has been underestimated thus far, just because short reads do such a poor job of identifying them. So here are two examples from recent publications. On the left, I'm showing a paper from the Jackson lab looking at isoform detection in HER2-positive breast cancer using bulk RNA sequencing. And they found over 140,000 isoforms in their data, and 67% of these isoforms were novel. That's what we're showing in the aqua. And what was also found is that these novel isoforms were enriched in oncogenes. There were 74 isoforms of HER2 alone, which is one of the best studied genes in cancer, so it really shows what remains to be discovered. And then also many of the novel isoforms were missing conserved functional domains or had different predicted protein localization, which indicates that these isoforms are likely performing an actual biological function in the tumor. On the right is a paper where they used single-cell RNA sequencing in ovarian cancer with long reads to detect both isoforms and fusions. So here I'm showing one of the novel fusions that they detected, which has IGF2BP2 on the 5' end fused to TESPA1 on the 3' end. And because an added issue with single-cell RNA sequencing is that you're usually only reading the 3' end of the transcript, what this resulted in the short read data is that all of the fusion transcripts were incorrectly assigned as overexpression of TESPA1 because it is the 3' partner. Conversely, if you look at the long reads, they were able to correctly identify that those reads were coming from the novel fusion, and the expression levels of TESPA1 here in the long reads are no longer elevated compared to normal cells. I'll switch now to talk about complete genomes, which include, as I said, not only small variants like SNVs and indels, but also phasing, methylation information, and structural variants. So here I'm going to show you an example containing the bottom three, so structural variants, phasing, and methylation. And I will also say that in the downloadable resources, we have material that covers performance on small variants also. So here I'm showing an IGV screenshot of our whole genome sequencing data. I'm going to take a minute to orient you to what you're seeing if you don't typically work with IGVs, since you will be seeing a lot of these in the rest of the talk. So here what we're showing are reads aligned to the genome, showing normal on top and the tumor samples below. And these are reads that align to the region surrounding the BRCA2 gene. The gray horizontal bars here are the reads themselves, and they are separated by haplotypes. So normal has 2 haplotypes and tumor has 2 haplotypes. And then the red and blue overlay indicates methylation status. So blue is hypomethylated and red is hypermethylated. So the first thing we can see in the tumor sample is a 29 kb region in haplotype 1 here, for which there are no reads aligning to this locus. So we can infer that there is a deletion in this first haplotype, which spans the BRCA2 locus. So we've lost 1 copy of BRCA2. And then the second thing that we can see is in the tumor is that the remaining haplotype, there is a tumor-specific hypermethylation of the region corresponding to the BRCA2 promoter. So in the normal cells, you see this is largely blue, and in the tumor, it is largely red. So this is essentially this results in decreased expression of BRCA2 from that second haplotype. So essentially, this is a complete loss of function of the BRCA2 tumor suppressor gene by 2 completely separate mechanisms. One is a deletion and one is methylation. And we're able to directly visualize both of those with haplotype information in the HiFi data. One more thing that I'd like to point out before we switch to our speakers is with cancer whole genomes, we recently released a somatic Whittle analysis tool, so that's what we're showing here, which you'll hear more about from our speakers later. And what this enables is that we now have a complete sample to answer workflow for cancer whole genome sequencing. So you start with DNA extraction with our nanobind kits to obtain high quality, high molecular weight DNA. That then goes into library prep with our SmartRel Prep Kit 3.0, followed by sequencing on a Revio, and then it goes into the somatic Whittle to do the analysis, which performs variant calling for all of the types of DNA variants I mentioned, plus a lot more, which I believe Lisa will touch on in more depth. So to close, all of that is to say that we're really excited about the things that PacBio is doing to enable tomorrow's cancer genomics by offering complete solutions for long-read whole genome and transcriptome sequencing. And we think this will provide the comprehensive and multi-omic view of cancer that we really need in order to understand it. Thanks. I'll switch to the speakers.
Thank you for the introduction and for the invitation to speak today. And thank you to everyone for joining. My name is Midhat Faruqi, and together with Dr. Lisa Lansden, I'll be presenting today's webinar, which is comprehensive variant detection in pediatric leukemia research with accurate long-read whole genome sequencing. A little bit of background about who we are. So both Lisa and I work at Children's Mercy Kansas City, which is a hospital in the Midwest that is exclusively dedicated to care of children. We have over 8,000 employees and almost 800 faculty, and we see around 250 new cases of pediatric cancer each year. One of the notable things about our hospital is that we have a freestanding genomic medicine center. So it was established in 2011 as the Center for Pediatric Genomic Medicine. And one of its notable accomplishments, this is now coming up on almost 12 years since this work was done, but it was pioneering the use of rapid whole genome sequencing for genetic diagnosis of patients in the NICU. And at the time, this was a Guinness Book of World Record, and it was from DNA to diagnosis in 50 hours. And the first patient that this study looked at was diagnosed with a lethal seizure syndrome due to a homozygous frameshift mutation in BRAT1. At the time, this would have taken weeks or months to do just through standard sequencing workflows. And so this was a rapid at the time whole genome sequence, though now, of course, there's places who can do this in less than 5 hours. The Genome Center then became part of our newly established Children's Mercy Research Institute, the building for which is shown here. It opened about 3 years ago, and I just want to take a little bit of time to point out the design. So these bands of light that you see here are actually supposed to represent nucleotide fragments in Sanger sequencing data as run on a gel back in the old days. So it's actually a high school project for students to come and figure out what mutation is represented by the data on the building. And then the flagship research project for our Children's Mercy Research Institute is Genomic Answers for Kids, and it's led by Dr. Tomi Pastanen. And the idea for this project was to create a first of its kind pediatric data repository for rare disease with a goal of sequencing 30,000 children as well as their family members for a total of 100,000 genomes. And with the idea being that this was to follow up on patients who had been undiagnosed in their disease journey so far. And so it was to take patients who had had maybe negative exome testing as well as negative microarray fish chromosomes, etc., and then apply newer, more advanced sequencing technologies and see if a genetic diagnosis for their rare disease could be obtained. So in that light, we began collaboration with PacBio in the fall of 2020. And since then, we've completed over 1,500 HiFi genomes of both probands and their parents, again, with a focus on rare disease. And the first 1,000 pediatric rare disease genomes done by HiFi sequencing were published almost 2 years ago in Genetics and Medicine, and that data is available. So one of the findings that came out of the study or was confirmed by the study was that genetic structural variation was much better detected by HiFi sequencing as compared to short read sequencing. And so what types of variants are we talking about? So this is basically looking at genetic changes more at the chromosomal level. So over several hundreds of base pairs, more in the order of thousands of base pairs or millions of base pairs. And it's looking at insertions, deletions, duplications, inversions, and translocations, again, at this level. For those unfamiliar with these types of variants, I'll just go into a little bit of background about them. So an insertion would be a piece of genetic material sort of being introduced into the new genetic sequence. A deletion is then a loss of the original sequence that's there. An inversion is a sequence popping out, flipping on end, and then reinserting it itself back in. Duplication, of course, is a part of the genome duplicating itself. And then a translocation is one part of the genome migrating to a different part of the genome. So maybe one piece of a chromosome going over to another chromosome. Now, what our GA4K study found is that HiFi sequencing provided the same level of detection, if not better, of small sequence variants or single nucleotide variants. And so that data is shown in red here relative to the same detection by short read whole genome sequencing. And then the gold standard for this comparison is microarray. And so even at this level, SNV detection was better than, had better sensitivity and specificity by HiFi than short read genome sequencing. But when you came to look at structural variants, those same deletions, duplications, inversions, and insertions, and then here the translocations are represented as break ends, there was vastly more, like on the order of thousands more structural variants that were detected, that were real and confirmed by orthogonal methods, by HiFi sequencing compared to short read sequencing. And then in some cases, this also offered better specificity. So you can see that break end detection becomes more precise with long reads. And this was one of the reasons why we started to think about applying this to cancer samples. Because if you are having thousands of structural variants not previously detected by short read sequencing being picked up in a germline setting, then it could be especially fruitful in applying it to detecting structural variation in cancer. So those same types of structural variants exist. And one example I'll give of a translocation is the well-known Philadelphia chromosome and the translocation between chromosomes 9 and 22, bringing the genes BCR and ABL1 together to create a fusion gene and then ultimately a fusion protein in chronic myeloid leukemia. But really, if you look at structural variation in leukemias in general, both acute myeloid leukemia and also precursor B-cell acute lymphoblastic leukemia, the classification of these diseases is really driven by their genetic subtype, and that in turn is defined by these different types of structural variants that one sees. And in certain cases, like hyperdiploid B-ALL, this can be relatively straightforward to detect. One can even do it with at the level of flow cytometry. But in other cases, these structural variants can be quite challenging to pick up. And because of that, we have a whole host of assays that we use in our laboratory to do this, such as FISH, karyotyping, microarray sequencing, as well as other methods, maybe fusion testing, in order to better define the genetic subtype of each of these leukemias. And obviously this takes a lot of time. It's also labor intensive. It's also expensive, which is why places tend to do this in maybe a tiered fashion, where whether or not you undertake a certain test depends on the result of the test that you are already doing. So that is where we wanted to explore the use of HiFi sequencing as a potential all-in-one assay. Maybe could we use it to compress all of these methods together into one? This would be especially useful in cases where there's a limited sample. So maybe an unsuccessful bone marrow or a hemodilute bone marrow or one where there was transport issues and specimen quality and quantity is more suspect. So our basic question was, could the application of HiFi sequencing improve this process? And so for that, we undertook a pilot project and selected 11 cases of pediatric leukemia. So these would be 7 B acute lymphoblastic leukemias and 4 acute myeloid leukemias. And 5 of these cases were known in that their genetic subtype was known. And 6 of these cases were unknown. So they had undergone our whole gamut of testing, and we were unable to find a genetic subtype for these 6 cases. So we selected the 5 known cases with sort of very classical rearrangements that one would see in our patient population. And then the unknown cases, we were curious if HiFi sequencing would be able to resolve any of them. And so to do this, we then took tumor and normal samples. Since these were leukemias, this is the neoplastic sample. It could be blood or bone marrow from diagnosis or relapse. And then for the normal sample, it was generally a remission sample. These were banked samples coming from our tumor bank. And a lot of them had existing DNA isolated already. And so another thing we were looking at is whether we could use existing DNA across that was isolated by a range of different types of isolation methods as well, and if HiFi would be compatible with that. And in this scenario, it was sort of hearkening to other technologies that maybe need primary sample, like optical genome mapping or HiC sequencing, where the use of those technologies isn't really compatible with existing DNA. So HiFi sequencing required about 5 micrograms to 7 micrograms, higher amounts if for lower quality specimens. And then we put each sample on one flow cell. So DNA from one tumor went on one flow cell for 30x whole genome sequencing with HiFi, and then same for the normal sample. And then the resulting sequence data were aligned and phased with PacBio's released human WGS workflow. And then we used a tool called Cerberus to analyze these data further. And this was very limited and focused in its initial scope. So we were just looking for break ends to see if we can find diagnostic translocations, as I mentioned. And we would do a manual assessment for any of these break ends that could be associated with cancer. And we were particularly excited to use Cerberus. So this was a tool that was developed by Dr. Kolmogorov and Dr. Keskis, who are at the National Cancer Institute, and we collaborated with them to help refine the tool and provide samples that the tool could be tested on. And it uses a series of different informatic techniques to call somatic structural variants on long read data. And then the announcement, this tool is available on GitHub, and the preprint describing its development and application were published on medRxiv 2 months ago. So on doing this, we basically found that all 5 of our known cases were easily identifiable by HiFi sequencing. So HiFi provided a way to do this with high sensitivity and specificity in certain cases like this RunX1, RunX1T1, that this was the only break-in that was called in this sample. Now, of course, the more cytogenetically complex a case is, then the more break-ins that there will be. But in doing this in a paired tumor normal fashion really helped us detect this with high specificity. We did not see a lot of false positive break-ins or break-ins reflected that were not detected by other methods like FISH or chromosomes. And I will show you this example in using an IGV screenshot. So this is basically representing the HiFi reads that were derived from this sample, and these have been aligned, and then we are using IGV to show if these reads map and where they map. And you can see that Severus is calling a break end in these two genes very cleanly in both RunX1 and RunX1T1. And if I zoom into the reads, you can see that part of the reads map to this portion of the gene and then break, and then it points to where they reciprocally map, and this is on chromosome 21 on RunX1T1. So it offered a very nice detection of this translocation. And then in certain cases, like this NUP98 rearranged AML, we knew that it had a NUP98 rearrangement based on FISH, but we did not have the partner gene defined. And with HiFi sequencing, we were able to define precise breakpoints in the exact partner gene. And so this rearrangement is known to be cytogenetically cryptic, so we could not see it on karyotype and needed FISH to pick it up. But now with HiFi, we could do the same thing as well as add additional information. And then even more excitingly, for the 6 unknown cases, just with this limited analysis, we were able to resolve 2 of the 6 with HiFi sequencing and then analysis for break-ins with Severus. And one of these was an AML case that we then found that it had a KMT2A MLL-T10 rearrangement. And the other was a B ALL case that had a ZNF3 84 rearrangement and in IGV. And so these are again the primary sequence HiFi reads, each individually represented here. And you can see that Severus clearly calls 2 break-ins, one on chromosome 12 and one on chromosome 19, involving the ZNF384 gene and the TCF3 gene. And you can also see that these reads partly map to chromosome 12, but then break, and the remaining part of the read maps to chromosome 19. And then reciprocally, the reads mapping on chromosome 19 map back to chromosome 12. So for these 2 cases that were resolved, they are known to be cytogenetically cryptic. So in the case of ZNF384, it's not possible to see on chromosomes. And this KMT2A MLL-T10 rearrangement can be visualized on chromosomes for some cases. However, when it is a complex rearrangement, it can also be cytogenetically cryptic. But here I wanted to take a moment to ask or to answer, in case you are asking, well, what does it matter if these 2 translocations were picked up? Well, for the AML case, looking at the World Health Organization classification of heme tumors, this is a disease genetic subtype defining rearrangement. So AML with KMT2A rearrangement is in its own diagnostic category. It is also prognostic linked to poorer prognosis, so this would place patients in a higher risk stage. It also points to its own treatment with the use of gemtuzumab, showing additional benefit in cases when added to chemotherapy in cases of KMT2A rearranged AML. And in pediatrics, this also puts this patient in line for a bone marrow transplant were it to happen in a clinical setting. And then the ZNF384B ALL case is also similarly in this other defined genetic alteration category. So it too is diagnostically defining. And then it again offers information on prognosis, which can alter the intensity of treatment that a given individual receives. So the next question that we looked at is why were these translocations not originally detected through the workflow and all the testing that had already been done for them. So to talk more about that and answer that question, I'll turn it over to my colleague, Dr. Lisa Lanston.
Thanks so much, Midhat, for a fantastic first half of the talk. Truly a hard act to follow. And I also wanted to briefly say thank you to all of those of you who are still listening. It's very appreciated, and also to PacBio for the opportunity to present this exciting work. We are so thrilled to be here. So for the second half, what I will be covering is these two cases that Midhat talked about. So why were they not originally detected? And then I'll also be walking you through some of the next steps that we have for our study. So to dive into the first case, this was an AML with a KMT2A MLT10 fusion. And through routine clinical testing, what we did first was cytogenetic testing for the karyotype. So what you're looking at here is going to be chromosomes and a chromosome study performed in-house. And what you can hopefully appreciate is that there are some differences between some of these chromosomes, particularly looking at chromosome 7. And so this is supportive of a neoplastic cell that we are analyzing. However, what's most interesting to us are going to be these two chromosomes here. So we're looking at chromosomes 10 and chromosomes 11, specifically looking for any abnormalities on this short arm, or 10p, where MLLT10 resides, and then this long arm, or 11q, or KMT2A resides. And when we went back and looked after we had resolved this case with HiFi sequencing, we weren't able to see anything suggestive of a translocation, telling us that this really is a cryptic rearrangement. So we also performed clinical FISH assay for this case, and what we're looking at here would be using two probes to determine whether or not KMT2A is rearranged. And so for the 5' portion of the KMT2A gene, or that first portion of the gene, we would be flagging it in green. And then for the second portion of the KMT2A gene, or that 3' end, we're flagging it in red. So here we're looking at a single nucleus that was observed under the microscope, stained in DAPI. And in that nucleus, what we're observing are 2 red and 2 green signals, and they're quite close together. So here, hopefully you can appreciate that these are relatively close, though slightly separated. Clinically, what we're expecting to see in order to call a true separation would be these signals on either side of the cell. And here they're nicely clustered, and so this would not be suggestive of a KMT2A rearrangement. What was really interesting to us when we used HiFi sequencing and then ran this case through the Severus structural variant caller was that we had these break-ends that were called, as you can see nicely illustrated by these blue bars in this IGV screenshot. And so what this is telling us, if we look down at the bottom track, is that there's actually a break within the MLLT10 gene, and that extends all the way through to the ARMC3 gene, where there's another break as well. Then we were able to determine through these data that actually there was this portion of chromosome 10 that was being inserted into the KMT2A gene in chromosome 11q. And so what I've done is I've created a couple of diagrams that can hopefully help make the prior data a little bit more digestible. And so what we're looking at is, again, this region on chromosome 11q that contains the KMT2A gene. And all that we've done here is we've turned the chromosome on its side, where now that short arm, or that p-ter, is going to be to the left, and then the long arm, or the q-ter, is going to be to the right. If we zoom into where our probes should be falling, we have that 5' KMT2A probe separated by about 40 kb, followed by the 3' KMT2A probe. Now 40 kb in a molecular space is pretty big, but cytogenetically, we would not be able to distinguish a difference between a 40 kb spacing or anything that's even a little bit larger than that, because it's just too close to tell with the eye under a scope. So here we said, well, what happens when we would insert this larger segment of chromosome 10 containing a portion of MLLT10 in between these two probes, and that would expand this spacing to approximately 1.4 megabases. Now again, that sounds rather large, but actually under a microscope in a FISH assay, that's not very big at all. And so we do not expect that we'd be able to distinguish a difference in the separation between these probes with just a 1.4 megabase separation. We're really looking for something more on the magnitude of about 3 megabases or more, though it certainly depends on the probe design. So the final question that we had, now that we have determined that this wasn't cytogenetically visible, is we wanted to determine whether or not there would be a functional gene product as a result of this insertion. And so what we're looking at here would be the 5' end of KMT2A. And then what's very exciting by using the data in this way is that we can determine the exact breakpoint within KMT2A intron 8, where there's that break and then joining of this inserted sequence of MLLT10, starting with intron 9, going all the way through the 3' end of MLLT10, including some intervening genes through intron 4 of ARMC3, and then we have that break and rejoin again into KMT2A intron 8 through the remainder of the gene, but then also through the remaining Q arm. And so what this tells us is not only do we have an insertion that we are able to detect through our sequence data, but we can resolve that yes, in fact, this is expected to form a functional product and result in that KMT2A MLLT10 fusion. Finally, one thing that we did want to do is follow up with an MLLT10 probe. So this is a probe that would be looking at whether or not MLLT10 is rearranged, since we now know that the 3' portion of MLLT10 was inserted into KMT2A. And sure enough, what we do observe is that we have this distinct separation between our 5' probe in red and our 3' probe in green. And we were really excited to see that this was confirmed with this orthogonal approach, really supporting that this rearrangement was real. So in summary, what I shared with you about this case is that our rearrangement was not observed via karyotype or FISH because it was cryptic for both our KMT2A probe and on chromosomes. Interestingly enough, MLLT10 is informative for this case, and so that is something we're starting to investigate on a research basis, is whether that might augment our AML testing. Also for microarray, it's a balanced finding, so we wouldn't expect to see this on microarray. And then although short-read exome was performed for this case, it was for SNV only, and so any structural variants were not assessed. And then unfortunately, RNA was unavailable for this case, so fusion testing could not be performed. Now, Midhat already kindly walked you through this particular case looking at the ZNF384 TCF3 fusion. And so I won't be going through this slide in very much detail other than to remind the audience that ZNF384 here on the left was showing evidence of a rearrangement here with reads that were mapping to chromosome 19. And here on chromosome 19, where we have TCF3, we had evidence of this rearrangement with ZNF384, which is found on chromosome 12. And so for this case, when we went back through the existing data that we had, what we saw is that it was also karyotypically cryptic. Now, this is not atypical for this type of fusion. It's known to be cryptic for karyotyping. Also for FISH, it wasn't included on our ALL panel, and so that's because the ZNF384 TCF3 fusion is actually quite a bit rarer than some of the other rearrangements in ALL. Then for microarray, this is also a balanced finding. So again, we wouldn't expect to see any evidence of this fusion necessarily on microarray. Short-read exome was also performed, but SNV only, so similar limitation to the prior case. And then interestingly, when we went back to the RNA fusion testing that was performed, we were surprised that it wasn't picked up in light of this additional data that we received through the HiFi sequencing. But it turns out that this was a bit of a pitfall of that particular fusion assay, largely because, again, this is a relatively rare rearrangement, rare fusion. And so neither gene was in that fusion panel, and is certainly something that we'll be considering in the future as we're doing follow-up testing on cases. So in summary, that nicely takes us through our pilot study. So again, to reiterate some of the comments that Midhat has made, what we're really excited about is that we were able to identify all 5 known translocations that we were expecting to see, and we were able to do so with base pair resolution and also determine the partner gene for the NUP98 translocation, which again is a cytogenetically cryptic finding. Then what was even more exciting for us is that for the 6 unknown cases, so again, these were genetically undefined by the technologies that had been used to attempt to bin them into a diagnostic category, we were able to resolve 2 using our HiFi sequencing. Both of these were cytogenetically cryptic findings, but that were fairly easily resolvable using our HiFi sequencing followed by SV calling. So for the remainder of the talk, I'll be walking you through our future directions. What are we doing next? So first and foremost, we're really interested in evaluating additional cases. We've already done some HiFi sequencing on a number of cases, some of which are genetically defined, some of which are still yet to be defined. And so I wanted to walk you through one additional example that we have followed up on so far, because I think it does a great job of illustrating just how good of a job this technology can do in resolving a complex case. So this case, interestingly enough, is a second AML case that has a cryptic translocation between chromosome 10p and 11q with the same players KMT2A and MLT10. So chromosomes in fish did not demonstrate any evidence of this particular fusion, and when we performed the HiFi sequencing, we were stunned to see this result. Now, I'm not going to dive into the nitty-gritty because this does get a little bit complicated, but what I'm hoping that the audience can appreciate is that on chromosome 10, we actually have 7 different changes that are occurring within chromosome 10p specifically, and then chromosome 11q, including a region that overlaps with KMT2A, we have 4 different findings. And so what we've done is tried to summarize them here, despite the complexity, in a way that's a little bit more easily digestible. And so what we're seeing is these red squares are going to indicate where the centromeres are for each of these chromosomes. So at the top, you're looking primarily at chromosome 10p, again, that short arm. And this is where we're going to start to see some of these different breaks in the chromosome 10p for a fairly complex event. Here, what we're looking at is primarily chromosome 11q. And so we're able to move through this chromosome until we're starting to see these 3 different events, within that 4th event being the majority of that chromosome being retained as expected. So what we're able to then do is determine why our FISH probes are not behaving the way that we're expecting them to. Why were they not translocating when we're able to observe through our HiFi data this KMT2A MLT10 rearrangement? Well, it turns out that there's actually a fairly complex inversion event as well that happens prior to the translocation. And so what happens in a nutshell is that we still have this green probe on KMT2A exactly where it's expected to be. However, because this inversion happens first, the break is going to be here, and it's going to result in a little bit of separation between our 2 probes, but they both will translocate to 10p. Then if we look a little bit further at the data, but again, not trying to belabor this point, we're actually able to detect various deletions that are occurring within these regions, though the true focus of today's talk is going to be on deep diving a little bit more into this translocation event. And so what this looks like in chromosome ideogram space is that, again, we have our MLT10 on 10p, we have KMT2A on 11q, and then we're going to have these breaks showing these different FISH probes for the KMT2A gene. So normally we would expect a separation here, but because we have that inversion event, instead both of those probes and the entirety of the KMT2A gene, though rearranged, is going to end up on 10p, whereas MLT10 is going to be split. MLT10 3' end will stay on the chromosome 10, and then the 5' end of MLT10 will move to chromosome 11. If we drill down even further, here's what we're able to tell. And we can look at our HiFi data in a lot of detail, and we're able to determine the exact breakpoint within KMT2A where this inversion is. We're able to see where it breaks outside of the KMT2A gene, causing that separation between the two probes. And then it will allow us to determine through the use of Severus exactly where that translocation breakpoint is, and show us that, in fact, this forms what is expected to be a functional fusion product, where we do have the red probe with that 3' end of KMT2A still present, which is why it's not detectable via FISH. But then we have the 5' end that's actually closest to MLLT10. We have a break within intron 10 of KMT2A, and then we have this break within the 3' end of intron 9 of MLLT10 through the remainder of MLLT10 gene. And so what we're really interested in seeing is whether or not we could actually detect a functional fusion product for this particular case, because we did have RNA available. And so what we did is we performed long-read Iso-seq sequencing, and what we were really pleased to see is that this very nicely demonstrated that we did in fact have a fusion transcript form. And so here what you're looking at is MLLT10 on the left, KMT2A on the right, and I realize it's a little bit difficult to see, but here we're starting to see KMT2A from the 5' end until a certain point where it all seems to stop. If we zoom in on this region, and I apologize because through IGV viewer it is a little bit difficult to see, but this is saying that we have chromosome 10 material that starts here, and it will extend. That's where it's mapping to. If we look over at MLLT10, we can see the chromosome 11 material, and that it actually carries through the remainder of the gene, with this Iso-seq then telling us that in fact we have that fusion transcript present. So in summary for this particular case, again, this was a cytogenetically cryptic finding both by karyotype and by FISH. Then by microarray, it's a balanced finding, not expected to see necessarily. Short read exome was performed, but it was SNV only. And originally RNA fusion testing was not performed, so not on a clinical basis, but then when we used Iso-seq on a research basis to follow up after doing the HiFi sequencing on DNA, what we observed is that sure enough, we are able to confirm the presence of this particular fusion transcript, which is really exciting. So next, what we want to do are a series of additional steps because we feel like we're just scratching the surface on what this technology can do. And so one of the things we're really interested in answering is how well would performing the tumor-only analysis work, especially in terms of structural variant detection. And so what we need to start to take into account would be the percent tumor that we are looking at, as well as limit of detection, depth of sequencing coverage that needs to be performed to determine these various events. And one thing that I'll note is that we did use relatively high percent tumors for all of our cases, and so we'd like to really push the limit and see how close to subclonal events we can find, and also do this with phasing with SNVs, for example. What you're seeing over here is that when we do tumor-only analyses, we really do see a significant increase in the number of structural variants that are called, and so we're hoping that by refining some of the analysis parameters, we might be able to move it closer and closer to when we're doing a matched tumor normal analysis, whereas that number of structural variants with Severus really does decrease quite a bit when we introduce that matched normal. We're also very excited to start to look into other genetic variant types. These are those that would be called by Severus specifically to start. And so these would be things like inversions, deletions, duplications, and especially looking at these for the remaining 4 unknown cases. So as an example, we've already seen in one of those remaining unknown cases that we actually are able to detect a amplification. This would be within the PAX5 gene. And so here you're looking at the tumor sample along with the paired normal, and we can very nicely see that Severus is calling that amplification within our HiFi data. And so we're really excited to pursue some of these additional findings further. Almost last, but certainly not least, what we are wanting to do is then layer on top some of these other types of changes that are called by the HiFi data. So this would be things like sequence variants, methylation changes, mutational signatures, et cetera, et cetera. And so here what we're able to see is all of the different components of the workflow that we can try to start to access. And we're hoping that we're going to be able to slowly but surely introduce these into our workflow overall, because it is such a powerful approach where we can capture copy number, mutational signatures, HRD, for example, and many, many more into the future. And so it's something that we're planning on tackling next. So then finally, what we would really love to do as well is start to expand this to other tumor types, including solid tumors. And so we've started to do those sequencing efforts, and stay tuned for those results. Then finally, what I wanted to end with is also a slide that Midhat had shared. So this is kind of parting thoughts on considering weighing pros and cons of testing assays, considering time constraints, monetary constraints, and also seeing whether or not we might be able to work our way towards an all-in-one test. We are really hopeful with this pilot study and the other data that I shared with you today, that some of these efforts, especially in the HiFi space, might march us closer and closer to the ability to reduce turnaround time, reduce additional tests that are necessary, though we realize that it will very much vary laboratory to laboratory, clinical space to clinical space, and patient to patient. But we do think that this is a really promising new technology and are excited to see where it leads. So with that, I would love to thank so many of the individuals that have made this work possible. So these are people at Children's Mercy as a part of the CMRI, also as a part of our cancer genomics team, looking at individuals in our Department of Pathology and Lab Medicine as well. A huge thank you to our strong collaborators at PacBio as well as at the NCI. And then biggest thank you by far would be to patients and families for their willingness to enroll in these research studies and allow us to do all this really exciting work that hopefully can come back and serve them. And then finally, would also like to acknowledge all of our funding sources that have made all of this possible. With that, thank you so much for your attention, and we'd be very happy to take any questions.
Awesome. Great, thank you so much, everyone. We're going to go through a few of the questions that are in the Q&A section here in the few minutes that we have left. Some of these have already been answered. Thank you to Midhat for doing that while we were on the call. But I'll read them out loud for everybody to appreciate. So the first, do you use tumor normal sample pairs to call structural variants using Severus? From my understanding, hematological cancers often lack paired normal samples.
Yep, great question. So we did for this study use tumor normal pairs, but Severus as a tool can handle both tumor only and tumor normal calling. These paired normal samples are harder to come by in heme malignancies, but for us, these were samples in our tumor bank. So we had remission samples we could readily use as normal sample comparators. But for other testing that we do, the normal sample, which is usually post-chemo blood or bone marrow following induction, usually becomes available in 1 to 2 weeks for most cases. We just wait for blasts to go to zero.
Awesome, thank you. Here's another one. Do you have a specific workflow you use for sample extraction, for example, the nanobind kits?
We have used nanobind and it works well, but I think one of the things that came out of this for us is that we used even archived DNA or stored DNA, some of which was isolated before the Revio existed. So it was really cool for us to see that you could use DNA isolated from a range of methods, QIAGEN, TKM, chemogen, et cetera, without even specific protocols or steps taken to maximize, long read DNA fragments and still get very nice and usable HiFi data from those. It was one of the strengths to this, I feel, because for other sequencing technologies or other molecular technologies like OGM, for example, you really need primary cells to do a specialized DNA extraction. HiFi sequencing would be similar in that regard.
Awesome. Here is another one. How about results on difficult to sequence regions? Were there any variants in that type of genetic locus?
Yes, though, as Lisa mentioned, we haven't fully analyzed these samples for all types of genetic variants yet. So we have yet to do full deep dive into methylation and CNV and small sequence variants, et cetera, et cetera. But we the KMT2A, MLL-T10 region that she was showing does have some difficult to sequence regions by short read. High GC regions aren't really an issue with HiFi, and there are multiple examples shared by a lot of the folks at Children's Mercy as part of the GA4K project. So Dr. Emily Farrow has multiple examples of cases where HiFi was able to detect sequences not seen in short read due to non-covered regions or regions that did not align because they were blacklisted in GRCH 37 or 38, et cetera, et cetera. So, but for these specific leukemia samples, especially maybe for those 3 or 4 unknown out of the 6, there might be a variant hiding in a difficult to sequence region by short read.
Awesome. I think that actually answers a couple other questions here that were asking about what about the remaining samples, so more variant types to look at there. Here's another question. You mentioned Iso-seq data towards the end of the talk. Can you talk about the value of incorporating the full-length RNA workflow into what you're working on?
Would you like to take that one?
I would love to. So that's something we're just starting to dig into. It's very much hot off the press data in our hands, but it's showing great promise. So we've had an opportunity to sequence a little bit over a handful of leukemic samples so far using Iso-seq, and we are able to observe the full-length transcripts and isoforms of multiple different fusions within these samples. And so we think that it's really going to augment and support what we're observing in the HiFi data. And I'm really excited to dig into it further. So stay tuned.
Next webinar. Okay, I think we have one more left. We're just a minute over, so I'll go ahead and ask it. Do you recommend any size selection of the DNA in order to do this?
Yeah, so that would definitely help. We typically share to a size of 14 kb with Megaruptor, and then size select on a Sage Pippin to fragments greater than 10 kb. But of course, as probably a lot of people are familiar here on the call, the HiFi sequencing works best in the 15 to 20 kb range of DNA fragment libraries. It's what maximizes the yield of sequence data. So size selection and sort of optimizing that definitely is the most efficient way to do HiFi sequencing.
Awesome. I think that was all of the questions that were in the chat. Oh, wait, one disappeared. Question to Lisa. Do you see that HiFi could replace FISH from the clinical perspective? Will the sample input be an inhibiting factor or the turnaround time?
Excellent question. So I think that with FISH, it's really going to be challenging to beat that turnaround time. Even if we would do a tumor-only analysis, there's still the prep, the sequencing, and the analysis. Whereas with FISH, if you know what your target is, you can turn that around within 24 hours, and that's a pretty tall order to do with NGS. That said, we are hopeful that we'll be able to do this in a rapid fashion, especially through a tumor-only approach, and we think that that's going to add on additional valuable information information for clinicians so that they can make those decisions a lot faster. In addition, I know you mentioned the amount of tumor or blast percentage, for example. That's something that we're actively investigating now, because we do know that there's going to be some of those lower level variants that are still very clinically important within these samples. And so making sure that we're understanding exactly what the tumor input needs to be, along with the depth of coverage, so that we can fully detect what we're expecting to and need to in these samples will be key.
Well, thank you so much to both of you. I really enjoyed this, and thank you to everyone for attending. Again, there'll be a quick survey coming after this, and we'll be in touch in the very near future with the recording of this, which you'll be able to watch on demand. Thanks so much, everyone.